Okay students, let us discuss another question, that is mechanism of HIV multiplication and how it leads to AIDS. Generally, you know, it's a very popular disease nowadays that is related to AIDS. Lot of awareness is created by the government now, you can see. So what is the action which is going to take place in our body, especially by the HIV virus, we will see now. Generally, once the HIV virus, which is called human immunovirus, which is going to get attacked to the particular cells, these cells are with CD4 markers. Okay, so the cells which are going to get attacked, especially what are that cells? CD4 marker cells are attacked by the HIV. So these HIV, human immunovirus, will attack CD4 marker cells. What are the CD4 marker cells? Generally macrophages, dendritic cells, and another one is TH cells. So these are the cells with CD4 markers. Why? Because HIV virus consists of GP120, glycoprotein 120 on its surface, which is perfectly fixed with CD4 marker. So that's why a particular tissue tropism you can able to see here. So HIV virus is going to attack CD4 markers and it will bind with this particular type of cells. Okay, after the binding, what is going to happen, we will see. Now this is the red color, you can see, this is all about HIV, which is going to fix it with the CD4 marker cells. After that, what is going to happen, we will see now. Generally, HIV virus consists single stranded RNA and reverse transcriptase. What is this? Single stranded RNA, single stranded RNA, and another one is restriction, okay, the particular reverse transcriptase enzyme. So like that, these are the two very important structures we can able to see in the HIV. Single stranded RNA, reverse transcriptase enzyme. Because of this particular enzyme, reverse transcriptase, single stranded RNA is going to become double stranded DNA. So now you see, this is single stranded RNA is going to become double stranded DNA with the help of reverse transcriptase enzyme. So after this formation, this particular DNA is going to incorporate with the host cell DNA. This is the host cell, means our body cell, our body DNA, which is going to get incorporated by the virus. So now RNA become DNA in the virus, and that DNA, means viral DNA, is now incorporated and attached with the help of ligase enzyme. What is this? Ligase enzyme. With the help of ligase enzyme, this viral DNA is incorporated with our DNA. Now you can see red and black, I am mixing both. So because of this particularly, there is a provirus. Now our DNA is under control of viral DNA. So like that, now this is become hybrid DNA because of the mixture. So this is named as provirus. This particular cell in which viral DNA is merged, this particularly this genome, this particular cell we can name as provirus. This provirus starts synthesizing mRNA. This is called transcription. So this particular cell releases mRNA. mRNA automatically, what mRNA is going to do? mRNA finally leads to translation, that is called protein synthesis. So like that, transcription occurs and finally what happens? Translation occurs. So because of transcription, mRNA synthesized, because of translation, proteins are synthesized. So these particular proteins are finally going to construct the material which is needed for that particular virus. So virus is slowly manipulating our particular genome and particularly synthesizing what it needs. So whatever the needs are there to that particular virus, so that is going to give small viral buds. So like that viral buds are released out. Viral buds are synthesized with the help of our cell. By using our DNA, the virus start producing virions. So it is working like a small factory. It is working like small factory. So like that virus production increases in our body. So like that it attacks macrophages, it attacks dendritic cells, and it will kills. Particularly it will kill TH cell. So like that in the TH cell this process is not happening. Remaining cells, this type of synthesis of viral buds we can observe. So slowly what happens? TH cells number decreases and decreases, and virus number in our body increases and increases. So finally TH cells number, which is very important for the recognition of pathogens, that particular TH cells are very low in the patient. He is called HIV patient. After 10 to 15 years, this type of condition will continue. When the TH cells number extremely low, at that time minimum small diseases will attack that particular patient. He is converted into AIDS patient. So like that the climax stages of HIV, when the people is susceptible for so many types of diseases, his immune system will become low. That particular condition, the climax of HIV is called AIDS. So like that HIV virus completely take over our immune system and dominates itself. So due to the failure of immune system, the AIDS patient is going. Okay students, let us discuss another question. This is sexually transmitted diseases from reproductive health topic. So generally the common diseases here it is given. So that's why you have to name the disease and you have to give the pathogen which is responsible for that particular disease. So here you see, here I gave 3 bacterial diseases, 4 viral diseases and 1 protozoan disease. Total 8 are there. In examination you have to write at least 4 without any mistake. So here you see, first one is gonorrhea disease. Gonorrhea disease is caused by Neisseria gonorrhea is a pathogen. Syphilis, Treponema pallidum. Chlamydiasis, what is this? Chlamydiasis. Chlamydia trachomatis. Chlamydia trachomatis. Next one is genital herpes or cervical cancer. Here 4, 5, 6, 7 numbers. Here these 4, 5, 6, 7 numbers are related to viral diseases. Genital warts or cervical cancer. Genital warts are also called cervical cancer, which is caused by human papilloma virus. Human papilloma virus. Next, genital herpes. Genital herpes is caused by herpes simplex virus. Herpes simplex virus. Hepatitis B. Hepatitis B is caused by hepatitis B virus. Same name. HIV infection and AIDS. It is very well known to you. That is HIV infection which is caused by HIV virus only. So that's why it is very easy. Last and final, that is protozoan disease, trichomoniasis. Trichomonas vaginalis. Trichomoniasis is caused by Trichomonas vaginalis, which is a pathogen. So sometimes the scientific names are very hard to remember. So that's why, for easy, I will tell you some important important tips how to remember easily. First you see gonorrhea. Here in the name itself you can see the gonorrhea. So that's why gonorrhea disease caused by Neisseria gonorrhea. So name itself indicates the pathogens hint. Anyway, Next one is syphilis. There is no hint for that. Why? Because the name is not relevant. So that's why, okay, you can, if you want, you can leave. Next, chlamydiasis. You see chlamydia. Pathogen name is reflecting in, okay, disease name is reflecting in the pathogen name. So that's why the similar reflection in Gonorrhea visa and same type of reflection you can see in the chlamydiasis also. Coming to the genital warts, cervical cancer, this is human papilloma virus. Next one is genital herpes. You see herpes. Kabatti this genital herpes is caused by herpes simplex virus. Kabatti that is very simple to remember the virus name. Herpes simplex virus which causes herpes disease. Okay, next one is hepatitis B. So this is very easy. Hepatitis B, HIV, that is leads to AIDS. Trichomoniasis, Trichomonas vaginalis. So this is also very easy. So like that, gonorrhea, chlamydia, genital herpes, hepatitis B, and HIV, AIDS, and trichomoniasis. These are the 6 diseases among the 8 you can able to easily remember. Okay students, now we are going to discuss about one of the important question and also very interesting question, that is all about natural selection by industrial melanism. There is a experimental proof is there for this particular question, so that's why it is very important. This is a practical proof operating natural selection. So how nature is important for the survival of the animals. So that type of point here we are going to mainly focus in this discussion. So here you can see, my dear students, here suppose if you see this is a tree bark, means tree trunk. Generally in a unpolluted area, okay, so if you go to village, okay, you observe where the industries are not there, in a unpolluted area, what type of color barks you can see? Generally the bark color is almost grey color. So like that, grey color bark is there in unpolluted area. So here there was 2 types of moths are there, that is called Biston betullaria. Betullaria. The moth which is called Biston betullaria moth, which is available in 2 forms. There are 2 species of this particular. One is melanic form, means which is in black color. One is in grey color. So like that, there is a grey moth and also there is a black moth. So 2 varieties are there in unpolluted area. Suppose if you see, okay, what is going to happen? So generally here you can see this is the moth. Okay, this is all about particular. You see that this is all about grey color. Suppose, so grey color moth. Okay, so here another moth I am drawing. So you see this is another moth. So one is the grey color moth and another one is, what about this particular? This is about black moth. So grey moth, black moth, 2 are there. So suppose a bird comes near to this. Okay, so in this, okay, a hunting bird came here near to this particular. Okay, which color looks first? Just you can guess which color looks first. Why? Because you see in the grey color moth, okay, grey color background of the bark, grey color moth do not appear. Why? Because it has camouflage. So because of that mimicry color, grey color is merged with grey color, so automatically the bird cannot able to detect that particular moth. So on the grey background, on the grey bark, black moth is there. So automatically this black moth is eaten by this bird. So like that, in unpolluted area, especially black moth's number decreases and grey moth's number increases. Why? Because in that area nature is supporting the grey moths. Same situation we can see in another condition. Let us discuss polluted area. Okay friends, let us discuss about the particular polluted area. You know, after industrialization, okay, after industrialization, especially in the very big cities and metro cities, you can see lot of soot is released, and that is lot of smoke is released. So because of lot of soot, what happened about the tree barks? That tree trunks, tree barks became black color due to release of soot. So because of that, the tree trunks became black. So in the black background, automatically black moths are protected. So in another area, we are discussing about polluted area, especially in the polluted area, black color background you are observing, especially the tree trunks are, which color? Tree trunks are turned to black because of the deposition of soot. So now this black color background is going to support black moths. So automatically on the black background, grey moths are easily visible. So that's why the bird is going to attack this grey color. So the birds are eating grey moths. So automatically black color number increases in the polluted area, grey color population decreases. So like that, one side environment is giving support to one particular type of species, especially due to coloration here we are discussing, and another area nature is supporting to another color. So like that, two different things we are observing, two different types of operation we are observing with the nature. So is it true? Is it really? This is experimentally proved by one scientist called Bernard Kettlewell. Bernard Kettlewell, scientist, he experimentally proved Darwin's natural selection theory. Whatever the theory we discussed about the nature, this is all okay, very popular theory which is called natural selection by Darwin is experimentally proved by Bernard Kettlewell. So he took same number of black moths and grey moths and released in a village called Dorset. This is unpolluted area, and Birmingham. What is this? Birmingham. So which is a city with industries. So polluted area. So same whatever we discussed. So equal number of black moths, equal number of grey moths are distributed in those particular areas, but after some days or after some months, he visited to particular village and also particular polluted area and unpolluted area. There in the Dorset, he got more number of grey moths. He got more number of grey moths. And in the Birmingham town and the polluted area, here he got more number of black moths. So like that, nature, how it is operating in particular area and how it is supporting to the species evolution, here we can see in this particular topic. So this is one of the important question. Okay students, now we are going to discuss about one more important and interesting question, that is all about ECG, electrocardiogram, electrocardiograph we can also call. You know what is its importance? Its importance is how the heart is working, the physiological actions, how the heart is doing, we can see by a electronic mission in the form of graph. This is all about ECG. So here in the ECG, there are most important events now we are going to discuss. That is all about P, you see this circle, and also Q, R, S, T. P, Q, R, S, T. The alphabets we will use here, P, Q, R, S, T. So here P is a complex, okay, P is one that is called atrial systole. Atrial systole. In the functioning of heart, so first atria contracts. So that event, atrial contraction event, is noticed especially because of this small projection. This is all about P. How much time it will take? It will take only 0.1 second. Okay, after that you can see a big complex, QRS complex. From here to here you can say that this is all about QRS complex. You should not say individually QRS. You should say QRS complex. So this is P wave, QRS complex, T wave. So like that, P wave, QRS complex and T wave. So wave, wave, in between complex. So like that you have to mention. So P wave, which is called atrial systole, which sustains for just 0.1 seconds, and the next one is QRS. So atrial systole, then immediately ventricular systole. Both will happen in 0.1 seconds only. But in the functioning of heart, we can see this ventricular systole. Generally, ventricular systole will take 0.3 seconds, like that it is written. But where that 0.2 seconds gone? Yes, that is gone because of this traveling. You know, if you see the structure of heart, especially, you know, in the structure, you can notice the particular SA node, AV node, bundle of His and Purkinje fibers. So like that, from SA node heartbeat starts and it will travel through bundle of His and Purkinje fibers, and finally contraction occurs. So like that, the traveling time is nothing but 0.2 plus 0.1. What is the meaning of this 0.2? This is all about AV node to bundle of His to Purkinje fibers, the traveling time. But real contraction happens in 0.1 second. So like that, this is the real contraction time, same as atrial systole, same as ventricular systole. Okay, so that's why QRS complex is together, we can call it as a 0.1 seconds of time this will happen. Next, after this contraction, slowly it enters into final relaxation. This is called cardiac diastole. In the cardiac diastole, how much time I wrote? Only 0.2 seconds I wrote. Actually, in examination, in the functioning of heart, how much time you will note? 0.4 seconds you will note. But here how much it is? 0.2. Where is that? That is the rest time. See, from here to here, the 0.2 seconds of time here, it is not mentioned. Why? Because it is the resting time of heart. So overall 0.6 seconds only the official time of functioning of heart. Why? Because SA node can able to generate 100 action potentials, especially 100 heartbeats per minute. But during normal conditions, it can able to produce only 72 beats. Why? Because the 0.2 seconds of time it is taking rest. So anyway, the T wave, whatever you are observing from here to here, this particular T wave is diastole stage. Between this cardiac diastole and ventricular systole, there is a gap you are observing. This is called ST segment. It is a zero voltage phase because there is no such action is going on. So that's why this is all about ST segment. So anyway, P, that is all about 0.1 seconds, QRS complex 0.4 seconds, T wave 0.2 seconds, 0.1, here it is 0.4 and here it is 0.2 seconds. Overall, if you see the intervals, from where to where you can see the interval P to R. This is called PR interval. PR interval will consist 0.1 to 0.2 seconds. Next, QT interval. From Q to T, QT interval, this is for 0.4 seconds. So we are including this T also. And finally, this is all about R to R interval. Suppose if you take two heartbeats. This is one heartbeat what we discussed now. Another heartbeat is there beside. So if you take two heartbeats, the peaks of the ECG, what is the peak? R. Here what is the peak? R. If you take two, and if you measure this time, this is all about 0.8 seconds. So in between we are also including the rest time. So generally we will say that heartbeat will take 0.8 seconds. So like that you have to mention by putting the seconds of time, and particularly for waves and also intervals.